Okay, great.
And we're live.
Good. So, hello everyone. Welcome to the final talk in our session. It is definitely my pleasure to introduce Steve Prescott. He's a senior scientist at SickKids. He's an associate professor in the physiology and biomedical engineering. And he does very interesting work in different stages of the neuronal hierarchy, going from the periphery to the brain, the central system, studying coding, studying both single neurons, axons, and circuits to try to figure out really the key aspects of information in our brain. So yeah, the stage is yours, Steve. It's a pleasure.
Thank you very much, Itai, and thank you to the organizers for inviting me and for putting on a fabulous show. I really enjoyed the presentation that I've seen so far, and I'm looking forward to the trainee talks after this. So I'll get straight on with my talk, so I don't go over time because I don't want to cut into the trainee talks. So today I'm going to talk to you about a number of sort of interrelated subjects. Sort of start off with a general sort of introduction to different coding strategies, focusing on the differences between spike rate and spike timing based codes. And then based on an interest in spike timing, we'll get into sort of the implications of precise spike timing, specifically with respect to synchronous spiking. And then I'll finish off by looking at the basis for precise spike timing and a number of different factors that sort of go into affecting precision. So with that, we'll get started on the idea of this neural coding. And so virtually all neurons rely on action potentials or spikes to transmit information to other neurons. And based on the fact that most spikes have a stereotypical shape and size, we'll very often sort of simplify this representation of voltage over time as a very simple sort of raster where each of these ticks represents a spike. And so you can look at a raster like this and ask, well, how do we want to characterize this? And I think, we can, simply count the number of spikes that the neuron produces. We can do that over a relatively long window. But if you might suspect that this rate is changing over time, then you might decide, well, actually, we want to try to capture that change in rate. And so we're going to use a narrower bin. And as you can see here, in fact, the rate is changing over time. But as you start to narrow the bin, you might ask, well, what is the appropriate bin size or the appropriate time window in order to count these spikes? And if you narrow the bin more and more and more, eventually you enter into a sort of a different regime where at most you'd have one spike per bin. And in fact, most of those bins are empty. They have no spikes at all. So at that point, you're not really counting spikes. It's more a matter of detecting spikes that occur in a short time window. You end up having, sort of 1, 0, 1, 1. And this isn't good, as I say, for counting spikes or for looking at the spike rate, but it is good for trying to capture patterns in the spiking. So the question is, well, what's the appropriate strategy, or what strategy might a neuron be using? What bin size should you be using to try to characterize the spike pattern of the spike rate? And a lot of that really depends on the sort of signal you might be dealing with, right? So if I told you that it was a slow signal responsible for the spike train that we're looking at, and in fact we have exactly the same spike train in these two examples, I think most of you would agree that using this wider bin is appropriate for capturing the fluctuations in the intensity of the slow signal. Whereas if you wanted to capture the occurrence of these very brief events and you wanted to actually resolve when those events occurred, then you'd want to use the much narrower time window and sort of resort more to a temporal code. But the thing about it is that the same spike train can be generated by these different stimuli, and the brain doesn't have direct access to the stimulus. All it knows about the stimulus is what the spikes are telling it. So could we actually look more into this and try to distinguish something different about what sort of response you might get from these two different types of stimuli? And so imagine that we gave the same signal but different noise to multiple neurons, right? So there's a common signal to multiple neurons, or to the same neuron over multiple trials. Is more or less the same sort of concept. And what you can see is that if you have a slow signal, then repeating that slow signal or giving it to multiple neurons, you'll tend to get asynchronous spiking, which is to say across these different neurons, the spikes don't line up very well. But the rate does co-vary. So all the neurons are firing at a relatively high rate, and then all of them slow down at around the same time. But that's very different from what you might see when you repeat this fast signal to multiple neurons. Here, you get highly synchronized spiking. You might miss a few spikes every now and then, but the spikes line up really quite well, right? So different coding strategies for different signals, and it's perhaps not surprising if you think about the Nyquist sampling theorem and things like this of what sort of temporal resolution ought you to be looking at. And this has been described before. I think sort of one of the most sort of famous papers describing the sort of patterning is Maiden and Sanowski 26 years ago now, where they showed that if you repeat a sustained stimulus, the first spike is really well timed, but then all of the other spikes, they sort of drift further and further out of phase, so you get very asynchronous spiking later on in the stimulus during the response. However, if you take exactly that same neuron and give it a relatively fast fluctuating current, then in fact what you see is trial after trial, all of the spikes, or most of the spikes at least, tend to occur at the same time and tend to occur with very precise spike timing and very reliably as well. So we're going to move on to the next part here, which is to ask, well, what are the implications of precise spike timing? So what I'm showing you here is that, okay, if you have a stimulus and it evokes a spike in multiple neurons, if the latency to that spike is fairly consistent, right, then all of those spikes are going to occur at more or less the same time. That is to say, with high precision, and across a group of neurons, that's going to lead to synchronous spiking, right? If for whatever reason, whether it's because of the stimulus or because of noise conditions or cellular heterogeneity, these latencies are much more variable from one neuron to the next, then you're going to have relatively imprecise spike timing. And at the network level, that's going to lead to asynchronous spiking. And this little graphic over here is just to remind you of the difference between precision and accuracy, where precision is really, referring to the tightness of the distribution. So all of these are really tightly, clustered together. And accuracy is more about, is that distribution centered on the bullseye? And as you can see here, well, there is actually a latency. You might say it's not actually very accurate, but it is in fact very precise. So that has implications now. If you understand some of these relationships, and I were to ask you again, okay, you have recordings from a set of neurons, and the rate tends to co-vary across those neurons, but the spike timing isn't, it's not very precise. The spikes aren't occurring synchronously. Then you might reasonably say, well, yes, this pattern of spiking across a set of neurons most likely was generated by some sort of slow signal, and it would be appropriate to then characterize that spiking using a relatively broad time window and try to sort of look at it from a rate coding perspective. On the other hand, if I'd given you these highly synchronized spiking, you might be much more inclined to suspect that it arose, that the network spiking pattern arose from a relatively fast signal, in which case you might want to resort to using a much narrower time window in order to capture temporal aspects of patterning within those spike trains. So I want to take you now to a real world situation to see how this how some of these concepts can be applied. And so what I'm showing you here are recordings from whisker primary somatosensory cortex, so essentially barrel cortex. And you're seeing responses from a number of different cells, a couple of trials from each cell. You're seeing the response to tactile stimulation of the whisker pad. So every time you see a little bump here, we're applying increasing pressure steps to the whisker pad. And what we did as a first pass is simply plotting a firing rate histogram, but we used a relatively broad Gaussian kernel, 100 millisecond kernel. So this is like using a broad time window in order to count the spikes. And what you can see is there's a spontaneous spiking, but every time there's a stimulus, that firing rate goes up a little bit, and the stronger the stimulus, the more the firing rate goes up, sort of, at the surface level at least, consistent with what you'd expect from a rate code. But if we take exactly the same data and now plot the firing rate histogram using a much narrower kernel, in this case 5 millisecond, what you see is something that's much, much noisier, but it's interesting that there are these characteristic blips and the firing rate in this narrow or high resolution firing rate histogram. And those blips seem to correspond to the onset and offset of each of these tactile stimuli applied to the whisker pad. And so what we did was just sort of arbitrarily apply a synchrony threshold, the idea being, that we're going to define all of the spikes that fit inside the firing rate histogram peaks that cross this threshold. We're going to define them as synchronous, and we're going to color them red. And every other spike that sits below that threshold, we're going to call them asynchronous and color them blue. So when we now color these rasters again, what you can see is the vast majority of spikes are blue, and their rate tends to sort of vary with stimulus intensity, as I sort of described before. But you do see this very sparse red spikes that occur at the onset and offset of each or most of the stimuli. And they're not necessarily there on every trial and every neuron, but they're enough that at the population level, we can resolve those onset, that transient synchronization. So I want to zoom in on just one of these onsets to give you a little bit better sense of what's happening. And what you can see here is the onset of the 10-gram stimulus is that this firing rate histogram that crosses our threshold, and then we say everything during this time where it's above threshold, well, those are synchronous spikes. And what you can see is that most neurons, they'll fire, in most trials, will fire a single spike. Sometimes you have two, maybe three, or none at all, but most neurons fire once. And this occurs within a very narrow window. So we're touching the whisker pad, and from about 6 milliseconds to 12 milliseconds later, virtually all of these synchronous spikes have occurred. And what I'm showing you here is a cumulative probability distribution of spikes for all of the different stimulus intensities. And what's interesting is that from the weakest stimulus intensity up to the strongest one, both at the onset and the offset, the latency, which is on average about 9 milliseconds, is completely insensitive to that stimulus intensity. So this is really a sort of a binary on-off. There's this transiently synchronized spiking, which just tells you something about the onset or offset, or these high contrast sort of edges that punctuate the stimulus. And if we look at the asynchronous spikes, on the other hand, you'll see that there's a lot of variability in the firing rate across these different stimulus intensities. But if you average them, as shown up here in the inset, you actually get a very good relationship between the firing rate and the stimulus intensity, consistent with rate coding. So just to make the point that rate coding and temporal coding are not sort of mutually exclusive. In fact, what we're trying to say here with this idea of synchrony division multiplexing is that you can have differential synchronization, and that the rate of asynchronous spikes and can encode one stimulus feature, like the stimulus intensity, whereas the timing of other spikes can encode different features, such as the contrast or edges or the onset or offset of a stimulus. So that's summarized here, where I'm showing you this idea that different spikes in the same neuron represents different stimulus features. So this is an example of multiplexing, the idea that, and that term came up this morning, but in a slightly different context. The idea here is using the same neurons, but different spikes to try to encode two different stimulus features, where the rate of asynchronous spiking will increase when the intensity of the signal changes, but you'll get this transient synchronization during these high contrast events at the onset and offset of the stimulus, if that onset and offset is abrupt enough. So now what I'd like to do is take you to look at the basis for this precise spike timing, focusing first on the stimulus properties, because I've already alluded to the fact that the kinetics are important, that fast events tend to encourage synchronization, whereas slow ones don't. And what I'd like to do is to try to give you a little bit of intuition as to what's happening. So what I'm showing you here are rasters now from a number of leaky integrate and fire neurons that are each receiving exactly the same sort of slow fluctuating noisy stimulus, but each of them is receiving independent noise. So the same signal, but different noise. And what we can do with the leaky integrate and fire model is that we formally want to sort of separate out what changes are due to the signal and what changes are due to the noise. So what I've shown you down here is we've actually converted the signal here and sort of depicted it as a reduction in the threshold. So when the stimulus intensity goes up, that corresponds formally to a reduction in the voltage threshold responsible for spiking. And then the voltage trajectory now is entirely driven by noise. So now we're seeing the voltage trajectory of each of, responsible for each of the rasters that I've showed you in the corresponding color above. And we're just zooming in on one area. And so what you can see is that when you have a slow onset stimulus, the threshold comes down, but the noise fluctuating around is causing these noisy fluctuations of the voltage trajectory. But each of these trajectories is smacking into the threshold at a different position, at a different time. And consequently, just sort of thinking about sort of arrival times, you're getting this, quite a broad distribution of the timing of these spikes, right? So the timing of spikes evoked by a slow signal are readily disrupted by noise, and that's essentially because the trajectories are hitting threshold at different times. Now, what I want to do is to contrast that with an abrupt or a fast signal. What you see here is this fast signal, which corresponds to an abrupt reduction in the voltage threshold. And now what you can see is, despite noise causing all of these fluctuations in the voltage trajectory, a lot of those trajectories, or all of the trajectories that are going to hit threshold, hit at nearly exactly the same time. A couple of those trajectories actually miss, and they don't generate any spikes. But that's notable in the sense that rather than generating a poorly timed spike, they don't generate any time at all. So this is sort of a reduction in the reliability rather than a compromise in terms of the spike timing precision, right? So under these fast conditions, the trajectories hit threshold at the same time. That makes for a tight distribution of the spike times, and in other words, precise spike timing. So again, this is very consistent with what I've shown you before in the Mainin and Sinowski paper, where these rapid fluctuations give you this consistent spike timing. And I would argue that it's more or less for the reasons that we've articulated in this simple simulation. Okay, now what I want to do is go back to this idea of multiplexing, but now instead of sort of these sparse intermittent changes in intensity, actually what I want to do is to consider a situation that's relevant for understanding vibrotactile input. So vibration typically is associated with a periodic stimulation between around 100 hertz, maybe up to 500 or 600 hertz. And it's been argued before that that this is encoded in somatosensory cortex in a multiplexed fashion, where the idea is the rate of spiking can tell you information about the intensity. So in this case, we're really talking about the envelope, or the peak-to-peak intensity of these periodic fluctuations in intensity. Whereas the period between that, or this is the frequency of the stimulation, this is captured in the spike timing, or in the interval between the spike times. So this is what's happening. I've sort of given you away the answer, but the question is, well, how exactly does that happen? And it's really maybe quite surprising, given how noisy the cortex is, that you could actually have precision that could help resolve stimuli at intervals of 2.5 milliseconds for 400 hertz. How can you resolve that? And let alone the fact that neurons, pyramidal neurons in cortex, won't fire above 100 hertz and things like that. So how is this achieved? So I'm going to show you some results from some simulations. But first of all, I just want to show you what would happen if you had a fully entrained response, which is to say one spike for every stimulus cycle. So we have 400, 200, and 100 spikes per second for the corresponding 400, 200, and 100 hertz stimulation. So that's fully entrained. But under these conditions, the firing rate is telling you about the stimulus frequency, and it tells you nothing about the stimulus amplitude. So that's not exactly what we'd expect, but these are unrealistic firing rates in the first place. So what we did is took an adaptive exponential integrate and fire model, and it sort of tuned it to respond more like what you'd expect for a neocortical pyramidal cell. And what you see now is that the neuron, all of them these neurons are firing or responding to all of these different frequencies at about 33 spikes per second. But they're doing so by responding only to intermittent stimulus cycles. So at the highest frequency, this neuron is skipping 12 cycles, or spiking on every 12th cycle, which is to say it's skipping 11 cycles. At the lower frequency, it's skipping 5 and responding on every 6th, and so on and so forth. But now you're getting the skipping, but all of them are firing at the same rate, so it's actually not telling you anything about amplitude either. But something interesting happens when you add noise. What happens is that the skipping becomes irregular. And you can see that in the ISI distribution that I'm showing you here down at the bottom, in the sense that this distribution splits into two small clusters. And those clusters are actually separated by the period of the stimulus, right? So if you have a 200 hertz stimulus, these clusters are actually separated by 5 milliseconds, which corresponds to the period for that stimulus frequency. And what's interesting is that these polar plots are showing you the phase locking, which is to say the spikes are actually occurring at a preferred phase that varies with the stimulus frequency, but it's actually quite precise. Despite the noise, the phase locking is preserved. And this is because the onset of these fluctuations are so abrupt that there's very little opportunity for the noise to disrupt the timing, but the noise will modulate the reliability or will modulate the likelihood of skipping. So you get this irregular skipping without compromising the spike timing, which I think is quite interesting. So we're going to keep the 200 picoamp signal on here and just show you what happens if we reduce this now to 100 picoamps. And what you see is that you simply skip more, and you'll get, on average, sort of about half the firing rate that we had for twice the signal amplitude. But the patterning is still there when you look at the ISI histograms, right? So under these conditions, these moderately noisy conditions, the firing rate does tell us about stimulus amplitude, and the spike pattern can tell us about the stimulus frequency. If we turn up the noise enough, eventually we compromise the phase locking, and that really sort of smears out and disrupts the patterning, so it doesn't work anymore. But there's a sweet spot, a sort of Goldilocks effect for moderate amounts of noise, and that actually corresponds exactly to what we see to the noise levels that would be expected in vivo. And this is just showing you some electrophysiology recording from a layer 2-3 pyramidal cell, and this is actually the same response that I showed you at the beginning of the talk. And although you might not believe it, there is in fact a very small periodic signal buried in this noise. And although you might not believe it, there is actually patterning in this spike train. And you wouldn't know that if you took sort of a gross ISI histogram, counted in relatively wide bins. But if you narrow that bin down, so we're now using a bin that's only 2 milliseconds long for this example of 100 hertz stimulus. So it's 20% of the period, stimulus period. And what you can see here is that it's very well separated at integer multiples of the stimulus period. And the phase locking is still very good. So noise is not always disruptive. That really depends on, the properties of the signal as well. So you have to factor that in. Now, I want to go forward now because I've been focusing on these stimulus properties, but now what about the neuronal properties? And you might have heard about sort of operating mode, this idea of integration versus coincidence detection. And the fact is that not all neurons respond the same way to certain types of stimuli. So I'm showing you two neurons here on the left. This neuron responds and spikes repetitively at a rate proportional to the stimulus intensity. And if you were to ramp up that stimulus intensity, the firing rate's simply going to increase. But that's very different from the neuron I'm showing you over here on the right, where this neuron only responds during abrupt changes in the stimulus intensity. And in fact, if you apply a ramp, this is a much steeper ramp than over here. If you apply a ramp, then in fact, you can see that there's a threshold rate of change of stimulus intensity that's required to make it spike. And so this is a good example of a neuron that we might refer to as an integrator. It's good at encoding stimulus intensity with firing rate, versus this neuron, which you might refer to as a differentiator because it's really taking the derivative of its stimulus intensity in order to decide whether or not to spike, but it's more commonly referred to as a coincidence detector. So here I'm showing you experimental data just running these two neurons through a battery of tests. You can see the integrator is a good rate coder. It's also very good at temporal summation. It'll add up these small inputs and eventually give a spike based on the integral of that input. But for the same reason that it remembers this past input, that makes it actually very bad at spike timing because it's not responding uniquely to the input it's receiving. It's partly responding to the input it received before. So that's in contrast to the coincidence detector, which is very bad at rate coding. It really has no sustained rate and is bad at temporal summation. But for the same reason it's bad at temporal summation, it's very good in terms of its spike timing. And so we can understand sort of how that might arise simply by doing some simple simulations. We start off with the same conductance-based model neuron, but we can reproduce all of the integrator traits by adding a subthreshold inward current, a sodium current, or maybe a calcium current that starts to activate at voltages just below voltage threshold. On the other hand, we can reproduce the coincidence detector traits by adding a subthreshold outward current or potassium current. Now, I don't have time to go into the details of this, but you should appreciate that neurons sort of exist along a continuum. And at this one extreme where we have, sort of the ideal sort of canonical integrators, the subthreshold voltage-gated inward currents really cooperate with input, with synaptic input, so that slow depolarization activates these voltage-gated inward currents, thus creating a positive feedback loop that encourages depolarization until a spike is initiated. And that's very different from what you see with the coincidence detector, where slow depolarization activates outward current, creating a negative feedback loop that discourages depolarization. So under those conditions, a spike occurs only if voltage threshold is reached before the outward current starts to activate strongly. Okay, and that means that you require rapid depolarization or a fast stimulus to get the voltage threshold before the negative feedback kicks in and stops you. Right, so that's what you're seeing here in terms of the integration, that the inward current will sort of cooperate and sustain depolarization, thus allowing good temporal integration. And that's reflected in the spike triggered average, or the average sort of stimulus waveform that precedes a spike. This is sort of extended and broad and monophasic. And that's in contrast to what you see with the coincidence detector, where the activation of this outward current is going to truncate the depolarization and sort of enforce a very short integration time. And that's reflected in a very short biphasic spike-triggered average. So you might be asking yourself, okay, so what does the typical pyramidal neuron do? And in fact, the answer is, well, it can do different things under different conditions. These are not necessarily sort of innate properties. They're in fact modulable. So under the sort of low conductance state, or if you were to patch on a CA1 pyramidal neuron in a slice and inject it with current, you'll typically see sort of repetitive spiking. But interestingly enough, if you were to simulate a high conductance condition, the same as you might see in the in vivo state where you have a lot of background activity, but we do that in an artificial way where there's no fluctuations. It's just a high conductance state. So reducing the input resistance by about 50%. What you see is that the repetitive spiking eventually fails, right? That it spikes more transiently. And if you look at the firing rate gain with respect to the mean, in this high conductance state, the pyramidal neuron becomes much, much less sensitive to the average stimulus intensity. On the other hand, if you now start causing fluctuations, this neuron becomes relatively more sensitive to those fluctuations than the neuron in the low conductance state, which is behaving more like an integrator. And again, that's reflected in the spike triggered averages, which we measured from these neurons. So just to say that, neurons aren't sort of necessarily one way or the other. They can actually be modulated along this continuum between integration and coincidence detection. And I just want to add one little bit here, which is the spike triggered averages. If you combine them, you can actually combine them in order to try to predict the cross correlogram. So this is the idea: whenever one neuron spikes, what's the likelihood of that other neuron spiking? So depending on what sort of input they're responding to, if they have a broad monophasic STA, they will tend to have a broad monophasic cross correlogram. So the co-modulation, the co-variation of the firing rate will tend to be on a relatively long time scale. And that's in contrast with coincidence detectors, where because of their narrow biphasic STAs, when you combine them, they actually have a multiphasic cross correlogram, which predicts very precise synchronization in a very short time window, and actually then quiescence, shared quiescence outside of those time windows. So sort of interesting, but I don't have a lot of time to go into that in more detail, because what I want to do is just sort of start to wrap this up by saying, okay, the coding properties, whether you're using rate coding or synchrony coding, depends on the one hand on properties of the neuron, sort of this integration versus coincidence detection, the expression of the different types of voltage-gated channels. But even within that, a given neuron might be modulated by other aspects of its network, sort of the state that it finds itself in. But this also depends on stimulus properties, whether you have slow signals or fast signals, which, if you think about the kinetics of the signal in terms of the synchrony or asynchrony of presynaptic neuron firing, all of this starts to get wrapped together. But even this, right, is sort of incomplete, because we also need to consider sort of circuit properties. I'm going to focus especially on the idea of feed-forward inhibition. And this study was actually prompted by a study that came out of Scanziani's group that claimed that pyramidal neurons have very steep input-output curves, and therefore, right, which we sort of see this here, if we stimulate the Schaefer collaterals, you reach a certain sort of threshold where the neuron goes from not responding at all to responding on every trial, and it's very steep. There's sort of no dynamic range, and therefore, for the network to have a reasonably broad dynamic range, if all of the neurons have steep input-output curves, then all of these input-output curves had to be staggered. Different neurons had to sit at different positions along this curve here. But that seemed to miss something that we were interested in, which is the effects of noise. So as I mentioned before, neurons in a slice condition in vitro are missing a lot of the noise that they would normally experience, a lot of the background network activity that they normally experience in vivo. And so we wanted to reintroduce that noise using dynamic clamp, and that's what I'm showing you up here, and then repeat this experiment. And what we saw is that when we added noise, lo and behold, this curve, for exactly the same neuron, same, otherwise the same stimulus conditions, it started to flatten out, and that sort of summarized down here, where you see like an 80% reduction in relative reduction in the slope of these input-output curves. We wanted a little bit tighter control, and so what we did is we resorted to dynamic clamp to simulate both the excitation as well as feed-forward inhibition with sort of the standard, kinetics and a slight delay to the onset of the inhibition. And then repeated this. So we're seeing pretty much exactly the same thing. This is some summary from multiple different neurons. But to cut to the chase, what you can see is that under control conditions, you do indeed have a steep curve. But as soon as you start to add noise, that tends to smooth out the curve. You can refer to that as dithering, or basically smoothing the nonlinearity associated with spiking and making sort of a much sort of more gradual recruitment of the neuron across a broader set of stimulus conditions. And that's really due to the noise, because if we do exactly the same thing with a constant shunt, that simply moves the curve to the left. It doesn't actually change its slope. But if we look here at some of the sample traces, we're doing this sort of as close as we could get to sort of the 50% response rate. What we can see is that when we add noise, we actually add a few spikes that are caused by noise. They're shown with a little asterisk here. And we're missing a few spikes, sort of by definition. That's also true here because we were sort of midway through. But what's interesting is that all the spikes that occur, all of the spikes that are evoked by a signal tend to occur quite, precisely with quite a constant latency, despite all this noise that we're adding. And so what we wanted to do is look at that in a little bit more detail. I'm going to draw your attention to this condition over here. What we're doing is simply showing you all of the spikes, from one neuron during this testing. And what you see is they're color coded, so the spikes that occur earliest have the lightest shade of blue, and the spikes that occur latest have the darkest shade of blue. And what you can see is that their timing really depends on variations in their voltage at the onset of the stimulus, which is at time 0 here. And that you can see under noisy conditions, that voltage really can vary quite a bit, and that impacts the spike timing, as you might expect. But it doesn't actually impact it a lot. We sort of go a spread, 10 to 90% sort of spread of 2.9 milliseconds compared to 1.4, 1.6. So it's maybe doubled the jitter, but that's not actually too much. But I want to remind you what I showed you before, that if we have a reasonably abrupt stimulus, then the noise modulated the probability of spiking without really disrupting the spike timing. And I showed you that also in the context of vibrotactile stimulation, where noise caused irregular skipping, but didn't disrupt the phase locking very much. So looking at this a little bit more, this is just showing you sort of these same input-output curves for a number of neurons, pretty consistent across neurons. And what you need to appreciate, though, is that this spike has to occur before the onset of this negative feedback, right? So you have this excitation, pretty abrupt excitation, and then it's going away, and then you have this inhibition that's kicking in. And that's actually very similar to what I told you about in the context of coincidence detectors, where they had a negative feedback on the basis of voltage-gated potassium currents. Now it's negative feedback on the basis of this feed-forward inhibition. But the idea is that the onset kinetics of the negative feedback, regardless of how it's implemented, can dictate sort of the properties of this high pass filtering. And I want to go back to this and just sort of explain this a little bit differently. Imagine you have a pretty abrupt stimulus, but if it doesn't go away very quickly, then the noise might still give you spikes that are quite delayed. But if you have this negative feedback kicking in, then what it's going to do is pull this excitation away, sort of, push the threshold back up, if you will, and prevent those certain trajectories from evoking a spike at all. So rather than evoking a poorly timed spike, they simply don't evoke a spike. But in that way, you can modulate the firing rate without compromising the spike timing. So it's sort of important to sort of put the reliability and the precision and try to modulate them somewhat independently. So it's not always true that noise is going to disrupt the spike timing if you set up the circuit or if you set up the cell in a certain way to respond to certain stimuli consistently. So to wrap up, I started by telling you about these neural coding strategies and try to really distinguish between rate coding and temporal coding, really as a basis of, well, how is it that you're counting the spikes? Are you using, high resolution or low resolution, a narrow bin or a wide bin? And really the choice sort of depends on, the appropriateness depends on the type of signals that you're dealing with. But if you do have precise spike timing, this is really a prerequisite for synchronous spiking at the network level. So precise spiking at the single cell level can give rise to synchronous spiking at the network level if those neurons are receiving a common input. And on the basis of that, well, the basis for this precise spike timing, well, it really depends a lot on the stimulus properties. So a fast signal, the spikes evoked by a fast signal are much more resistant to noise. And that's in contrast to spikes evoked by a slow signal whose timing is much more susceptible to disruption by noise. So the stimulus properties are indeed very important. But we also need to look at the neuronal properties. So this idea of integrators and coincidence detectors, it really is a continuum, but neurons that have voltage-gated inward currents that create a positive feedback loop, well, they're good at integration, but that is not necessarily ideal for spike timing, whereas coincidence detectors with this negative feedback that truncates the depolarization, that will tend to encourage this precise spike timing, exactly the same way that you tend to see with feed-forward inhibition. So this negative feedback, whether it's negative feedback on the basis of a microcircuit motif or negative feedback on the basis of certain voltage-gated potassium channels, they really serve as a quality control mechanism that ensures that a spike occurs before the onset of negative feedback, or that it that spike doesn't occur at all, which is a good way of modulating the firing rate. And so with that, I'd like to acknowledge the people in my lab currently, the past people, as well as some of the collaborators whose work I've told you about. I've highlighted some of the people throughout the talk, but I'd like to thank everyone for their hard work, as well as for the funding that we've received. And I think we have time for a few questions. Thank you very much.
Thank you. That was really interesting. All right, we have a few questions. Let's start with Spandans and Gupta. When in the regime of the temporal code, can you think of spikes as some kind of a Poisson process?
Well, at that point, you can look at the interval between bins that included spike and ask if they are truly random, if they follow the statistics that you'd expect from a Poisson process. Certainly if they're actually being driven by a periodic signal, then you'll find that they're not Poisson distributed. I think so I think it depends on what they're responding to, but looking at them with a narrow bin or just the interval between them is you need to have that resolution to really answer that question.
All right, a question from Maurizio. What could be the mechanism whereby the downstream neurons decode the multiplex signal?
Yeah, so this is where we think that maybe you can transmit the multiplexed representation across however long you might want to do that if you're using neurons that are sort of operating in the sort of midway between integrators and coincidence detectors. But if you then gave that signal, let's say, to a downstream neuron that's behaving as a strict coincidence detector, that's behaving as a very high pass filter, then it can pull out the precisely timed spike and not respond to the other ones. Whereas if you pass this, well, most of the spikes are already asynchronous and just responding to rate. But if you pass it to a very good integrator, it would sort of tend to filter out or neglect the precisely timed spike and pull out the rate code. So this is also sort of important to consider, what is the downstream decoder? And this is where some of those intrinsic properties at the cell level or, microcircuit properties can sort of implement filters that would help you pull out one or the other signal from the multi. So essentially to demultiplex that representation.
Great. Question from Scott. Do the noise results fit with the hypothesis that noise heterogeneity increase the information capacity of neural circuits, but at this single neuron level?
Sorry, what was the last part about the single neuron level?
So the idea that it's done essentially at a single neuron level, this increase of information capacity, like what you found with the noise, right?
Yeah, I think here so I think I think the nervous system has evolved to deal with noise, right? That getting rid of it would have been difficult, if not impossible. And so, and in some ways, rather than just dealing with the noise, it's capitalized on it. So I think it's true that it will cause variability, which for instance, if you think about the vibrotactile, the multiplexing of the stimulus amplitude and frequency, you really need that irregular skipping, which really relies on the noise. So that really connects up also with this idea of a broad input-output curve or smoothing that nonlinearity. So that really benefits from the noise. Now, on the other hand, you can't really do much with a single neuron, or at least a single neuron on a single trial, because when you have this probabilistic spiking, you don't really know what the distribution is like unless you're sampling across multiple trials, or more relevant for the biology, across multiple neurons receiving that same signal. So I think a lot of this doesn't really work until you've embedded that noise in amongst a number of neurons, and that's also assuming that the noise is relatively uncorrelated across those neurons, that you actually get benefit from it. So I hope that answers the question. If not, I'm happy to follow up with
That's good. A question from Taha. Is the change between the integrator and coincidence detector modes happening in a phasic manner for the pyramidal cells?
The change, so it's interesting. So this goes back to some of my postdoctoral work, actually, and you can see the example down here. Actually, it's an interesting nonlinearity which happens where increasing the shunt actually has the effect of increasing the voltage threshold a little bit, and by changing that voltage threshold actually occurs in a sweet spot on the, where the activation of M-type potassium channels or Kv7 channels occurs, and so you actually end up getting a more more sub-threshold or more peri-threshold activation of these Kv7 channels, which strengthen the negative feedback and sort of in a nonlinear way, sort of slow down or truncate, reduce the likelihood of voltage getting to threshold if it's going slowly enough that the M current has time to activate. So it's a very nonlinear sort of process, that back in the day, we were doing the sort of nonlinear dynamics to try to capture exactly what was happening. So it's sort of a very, it's a continuous thing that depends on the magnitude of the shunt combined with the magnitude of the M currents that's there, and then it's actually quite nonlinear when it starts to happen. So.
All right, I think we'll wrap up soon. I'm just gonna squeeze one question from Alex. Sure. Do you think inhibitory neurons exhibit analogous coding strategies?
Yeah, they always get short shift, but I think I think, well, inhibitory neurons have a great diversity, so the fast spiking ones versus the slower spiking ones that look more like pyramidal cells. I think it really, I've not investigated them, but I think they're probably responding to, they might tend to be engaged by different types of stimulus features or different types of network activity, which I think is very interesting. But I think traditionally they've gotten less attention in these sorts of investigations. So I don't have a good answer to that.
All right, good. So we are done. Andrea, you can take over. Thank
Thank you
you
very
again
much.
thank you, Steve.
Thank you so much, everyone. Thank you, Steve, for your great talk, to Itai for moderating, and to you for your attention. Please join us for the trainee spotlight session.